All right, so some of the examples that we would give in class is the idea. Let me take roll 0, 4, 9. Okay. Is it just Noah that's gone? Okay. So one of the aspects that we sometimes refer to is if you were helping a younger student or maybe a relative, for instance, if you were, let's say, helping out with DNA replication. Yes?
This is what you saw, right?
Well, he had that, then he changed, now he put it back. So
But that hasn't even been erased.
it you can see the D on there a little bit. But I think.
Well, he handed it in to me like this.
But that's right though.
Okay, yeah.
But yeah, I just wanted to be sure.
Saying that he changed this.
Yes. But I mean, some of the other players had seen him do that, so and kind of thought it was kind of funny. I think so.
Okay.
But it was taken care of, and I'm glad we're on the same page with that. Yep. All right. So, as we look at what we call DNA replication, okay, what steps are involved when it is DNA replication? What's the first thing that happens? It's the last thing we talked about yesterday. That's part of it, but that's too far down the road. What happens first? Okay, so keep in mind, okay, and it's hard to visualize this in 3 dimensions, but you had this double helix that wraps itself around like this. What happens to that double helix? It unwinds and then opens up. So when that opens up, what is it that is moving from one side of the double helix to the other? It's an enzyme. Say that again. Helicase enzyme, DNA helicase. What is that DNA helicase doing? Yeah, it's splitting the two molecules apart because DNA is known as a what type of helix? Double helix, okay? So as that DNA helicase is moving down the chain or the replication fork, it's breaking hydrogen bonds. As it's doing that, what's floating freely inside the nucleus? Other what? No. Nucleotides, they're floating freely in the nucleus, okay? In case you're maybe having a hard time following along, this was on page 201, okay? And as that enzyme breaks that apart, helicase is moving up the double helix or the replication fork. Nucleotides floating freely in the nucleus start pairing up with their complementary base pairs. Okay? And then what comes after that? Okay, again, it's the last thing that we had talked about. Doesn't it look like this? Yeah, it's hydrogen bonding. That will reoccur again, okay? So they begin to assemble in the proper sequence, okay? And then the second enzyme, DNA polymerase, will cause the hydrogen bonds to form again. And a lot of you have this diagram, looks like, written down somewhere on your paper, okay? So, what we want to do is just make sure that we can put that to use. And that is as follows, okay? So on here, okay, you tell me, okay, What is this guy right here? It's the helicase enzyme. You can say that if you want, because how many helicase enzymes are there? Just one. Okay? The reason we want you to be specific is because there's two polymerase enzymes. There's one for DNA and there's one for RNA. Okay, so we have this DNA helicase that is breaking these hydrogen bonds. Okay? So what are our base pairings as we go? What bonds with thymine quickly? Okay, and then we'll just stop here because these bonds haven't been broken. That's what those dots are designating, is hydrogen bonds. Okay, so now what we see happening is if we take this blue color, we said that nucleotides floating freely inside that nucleus are going to start doing what? Pairing up in the correct order. Okay, so they're again bonding with thymine is adenine, and then thymine, and then guanine. So you get the idea that the copy that's on the inside is the same that's on the outside. Does anyone not follow that? Is our left outside, we understand this so far? Okay, left middle, right middle, okay, and right outside. All on the bus. Good, 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 good. So what we see happening is we said, make sure you know this. Where is DNA found only? Only in what structure? Nucleus. So if you backtrack to page 197, okay, what is that illustrating? It's showing you the double helix in figure 10-6, okay? Then where's that DNA say it's coming from? Well, the nucleus, okay? And that DNA is located on what structure in the nucleus according to page 197? Chromosomes, okay? So some of you have maybe heard of independent, let's say genetic, I wouldn't say companies, but I'm trying to think of the proper word. Have you ever heard of 23andMe? Does that name, you're nodding your head yes. What would 23andMe possibly do? What use would you have for 23andMe? That's exactly right. You're finding your percentage of what it is your ethnic background would possibly be. Do we just make up the number 20? Why did they pick 23? 23 chromosomes has something to do with it because we as humans have how many? 46, but where do we get those 23 pairs from? One from mom, one from dad. So that's why it's called 23andMe. All right, so as we look forward then, we're going to start talking about RNA replication, okay? So what we see here. Oh, and then finally, yes, step one, helicase breaks the hydrogen bonds. Step two, you have nucleotides that are just floating freely inside the nucleus. So maybe at one point, you know, it's just hanging out here in the nucleus. They eventually just start pairing up with their complementary base pair here, okay? And then, what is the final step? What is this enzyme here that's forming the hydrogen bonds between these two? Don't just say polymerase, you need to be a little more specific. DNA polymerase. Okay, so that is where we had left off, okay? So, with this flow of genetic information, we call that transcription. And the example that we gave is, for instance, our quarterback, okay? Goes to the sideline. The sideline is the nucleus, okay? And the coaches are DNA. And then in this process, messenger RNA is your quarterback leaving the sideline and coming to the huddle, okay? That's the example we gave trying to make sense of this. Because back in, has anyone ever heard of the feudal system? That's something from world history. Do we know what the feudal system is or was? Some of you are nodding your head yes. Okay. So back in that time, could everyone read and write? No. So what might a person be called that could read and write? I don't know if you'll know it. You ever heard of a scribe? So a scribe is a person who could read and write. So when we look at this process of transcription, it's kind of like a scribe back in medieval times looking at information and reading and rewriting that. Okay, so there's a process of transcription, and then there's a process of translation. And all of this put together is this here, protein synthesis. Okay? Cole, you can come over here and wave. Wave to the boss. You want to say hello? No? Okay. Save that for later. Okay. All right. People didn't bust the irrigation lines, did you? Or is he just, maybe one of the heads isn't turning out there? Or maybe that's a valve box. I don't know. Oh, yep, that's a valve box. Okay. All right. He's doing your job. Maybe you want to go out there and help him. Yeah, earn some extra money. It's better than sitting in here, isn't it? Okay. Yeah, there's something to be said for honesty. Okay. Now, one of these things on the next slide, you're going to have to wrap your head around this. Do I need to back up? Okay. So if an irrigation head isn't turning, how do you try to fix that situation? Oh, that's a little more advanced than what I would try to do. I just step on them. You ever done that before? Yeah. Then it pops back up and whatever's in there might be flowing out of there. Oh, okay. Oh yeah. Now all I saw was the pickup. Now I see the air compressor behind there. Okay. All right. So we want to do one more slide, and we're going to make some sense of this. You need to wrap your head around this. Notice we are not talking about DNA anymore. How do we know? Because we're talking about RNA up here. It's still in the process of DNA replication, okay? But when we're talking about RNA, thymine is not found on RNA. For some reason, the chemical makeup is different, so uracil replaces thymine. Okay? So when you look at these three lines, this one, and this one tells you all the different aspects that makes this different than DNA. One, it's got the sugar ribose. Two, probably the second most important one, if not the most important aspect, is uracil is present instead of thymine, and that RNA is only single-stranded. PVC tubing used for irrigation, or is it that red and blue? Or that? Okay. Okay. You have pretty good luck changing heads, because I've had to do that before too. Sometimes they leak, and then you got to replace that head too. Okay. Does everyone have this information that wants it? Yes? Okay. So, what we want to do then is we're going to shut this off. Okay. How many of you are looking at this information and thinking, yes, this somewhat sounds familiar, yes, it somewhat looks familiar, or is this going beyond what you learned in elementary and middle school? Yes? Maybe a little bit beyond. Okay. So, what we want you to do now, okay, is turn to page 205. Okay? Look at figure 10-13. 10-13 on page 205. So if those headings were not on there where it says DNA and RNA, okay, how would you know that the right structure, the one on the right, is DNA? What clues do you see on there that tells you that that is DNA? The first thing is what? Yes, it's a spiral. It's a double helix. Okay, that's the biggest clue that that's DNA instead of RNA. Now, when you look at the left side where it's RNA, what sticks out in your mind that that's RNA and not DNA? It's what? It has uracils 1. What about the strand? Well, true. It's a single strand. That's right. Okay? So what you see further down on the line on page 205, down here, we're showing you it's just a straight line here on page 205. When it comes to messenger RNA, there's just a couple things that would change. Remember, we had said on previous information or previous slides is the idea that, for instance, okay, so football players, football guys, this is DNA in the nucleus, so that is, what did we say earlier? Close. The coaches, that's right. So we'll just put Mr. Williamson and then Mr. Mertz or Eminem. So the directions that is needed to get to the play, which is away from the sideline, has to be encoded in a message. The message comes from the nucleus or from the coaches. So if we are now talking about copying this on a template of messenger RNA, okay, start telling me the base pairings. Now be careful, don't just start spouting out nucleotide pairings without thinking first, or else you're going to make mistakes, okay? And we say mistakes, 1, try not to repeat them, and 2, try to learn from them, okay? So, what bond, now, we're talking about DNAs here, messenger RNA is on the outside here. So, on the outside, what's complementary to thymine? It's still adenine. So, what's complementary to adenine? Thymine, but think again. You're going to, if I just tell you, you're going to say, God, I knew that. You just said it no more than 2 minutes ago. Why is it uracil? RNA does not have thymine. The chemical makeup is uracil instead of thymine. So it goes A, then U. Now what about cytosine? Now everything else is just the same. So bonding to cytosine is guanine, cytosine, again, guanine, cytosine. What now? Do we put a U here? No. We put a what? Put an A. Then an A here, we're going to put what here? Uracil. Is anyone confused up to this point? We're looking at this and saying, we're saying all these letters, C and G and U, and why is it U here and not a T? Yes. Only with what we've done the last 30 seconds. Because in the nucleus, it's right. So again, in the nucleus, and this is DNA, so it's still thymine. If it's RNA, the uracil replaces thymine. Okay? Now, the reason something like that is so important, okay, is as we look forward to page 207. Be the first one to find the three-letter base pairing of AUG and tell me why that is so important on table 10-1. Exactly. And the reason that it's a start is the specific enzyme is looking for this segment on DNA. Okay? Because, remember we said we were going to try to crack open these this biochem book. Okay? So, the applications for that are as follows. When it comes to this entire process is called what synthesis? Protein synthesis. Okay? So this is coming straight from the author of Ira Bly and George O'Dane. Okay? It's a textbook of biochemistry. So there's rhymes and reasons on why we do this. Some hereditary diseases that could result from mutations inside of here could be something, some of these you had heard of. Albinoism, what might that be? What's, if someone, okay, they don't produce any melatonin in their skin, and that melatonin does what to the skin? Makes it darker, that's right. Okay, so albinoism, it's tyranase is deficient and melatonin is not produced, or yeah, I said melatonin, sorry, I meant melanin is not produced. It protects the skin and the iris of the eye against UV damage and skin cancer. So tyranase, what did you notice? This also ends in ASE, helicase, polymerase. Those are specific enzymes, okay? Amino acids make proteins. Proteins are classified as enzymes. So if these are not assembled correctly, deficiencies can occur in the human body, like albinoism. Has anyone heard of what would CF possibly be? You probably have heard of it. Cystic fibrosis, okay? That's a viscous mucus in bronchi passage leads to pulmonary bacterial infections. Inadequate lipase. So lipase must be a what? Ends in ASE. Just like helicase, polymerase. It's a specific enzyme. That's right. So inadequate lipase and bile salts produced by the pancreas result in malabsorption of lipids and fat-soluble vitamins. Okay, we'll go a little further into some of these disorders, perhaps later down the road. Okay, but just keep in mind the big item projects that we want you to know is what DNA replication is. It's the process of DNA helicase breaks hydrogen bonds. Nucleotides that are floating freely in the nucleus start to pair up with their complementary base pairs, and then you have DNA polymerase that puts the strand back together. Okay, and two differences between that of DNA and RNA. What would they be? RNA is single-stranded, and what else? Uracil instead of thymine. So when you're looking at that table in your book on 207, yes, all of these different three-letter base pairings that you see are coding for the amino acids that make proteins. And this is why in this protein synthesis then, when it's not done correctly, some of these medical disorders can be found in humans, whether it's cystic fibrosis or that of albinoism. If someone has hemophilia, what might that be, do you think? Yeah, because hemoglobin is what is the chemical makeup of blood. And if they have hemophilia, they're missing a protein to have their blood clot, which can be obviously quite dangerous and what can be life-threatening. Okay, and again, we'll get into some of that at a later time. Is anyone looking at this and thinking, my gosh, this is awful? Okay, and please try not to think that. This is just new information. This is perhaps new to you. Any questions? Okay, we'll pick up with this information then tomorrow.